We're going to talk about the growing diversity of H3N2 influenza A virus in swine. There's a lot to talk about, and then try to bring this back around to the impact this diversity is having both in the swine population as well as at the human-animal interface. So we know a lot more about the influenza viruses that we have in pigs in this contemporary period because of the USDA surveillance system. And it was implemented in 2009. It really began as kind of a pilot project before the pandemic emerged, but we had all the pieces in place that we were able to rapidly get it going through the USDA NOM system and interactions and funding from CDC to begin with, and then some of the supplemental pandemic H1N1 funds that will probably run out in 2015. I think Dave is sitting out there trying not to think about that, but it is a temporary program at this point, and hopefully we can see the benefit of this system and keep it going once the temporary funds run out. So there are 39 participating non-labs, so even though this is the USDA system, it really relies on those veterinary diagnostic labs like Iowa State and Minnesota and many other key diagnostic labs that the submitters were already submitting samples to, and we're just kind of pulling out of that system that was already in place and that expertise that's already there. So the USDA system uses a matrix flu A screening PCR, and the PCR positives can go for the hemagglutinin and neuraminidase subtyping PCR. Then those virus isolates go to NVSL and are forming a virus repository that can be used by researchers or vaccine developers, so that repository is there for public use. And then we've also seen a lot of growth in the number of sequences that get deposited into the public database GenBank through this system, with a priority on the hemagglutinin, neuraminidase, and matrix gene. But NVSL's also been doing quite a number of whole genome sequencing, and those sequences get deposited into GenBank as well. So again, this is a passive surveillance system. The primary source of samples are from diagnostic samples, so cases of sick pig with respiratory disease. And it's also a voluntary and anonymous system, which is very important to the swine industry, but it also brings some, I guess, unique challenges when you are trying to figure out what's going on with the viruses and how they've moved from one place or to the other. But it is anonymous, and the only identifying information that goes with these public sequences would be in the strain name, and that would have the state of origin of the virus. So although the vast majority are coming through the diagnostic stream, we do have a small number of viruses that come through the system that are associated with a zoonotic event, and probably the best example would be from these fair venues where we've got sick pigs and sick people, and those samples get submitted directly to NVSL. So what we've tried to do with the USDA system and what we're trying to move toward is a more systematic approach to monitor influenza A in swine. And this isn't just happening in the US. There are these initiatives in other parts of the world, but I think by far we've contributed the most to the public knowledge on influenzas and what's publicly available at this point. So again, it's about maintaining a virus repository, so you can go back in after you've analyzed sequence or analyzed the antigenic properties and pull these viruses out of the repository, either going through whole genome sequence or gene by gene sequence. And then for my lab, we're really interested in these viruses to look at the antigenic relatedness of these viruses, and can we predict this based on the genetic analysis, and then doing the research studies, whether that's looking at the pathogenesis of the virus properties in the pig or doing the vaccine studies with the relevant circulating strains. So we've been doing this surveillance system for a couple of years now, and we're at a point where we've got enough data that we can start to look at the trends that have happened from region to region, within seasons or between seasons and from year to year. And so what we've been looking at are the trends in the HA and NA. We also are looking at the matrix gene as a surrogate for reassortment with the pandemic virus, because as I'll get into a little bit later, we've seen a lot of reassortment between what was endemic before the pandemic and then with that pandemic H1N1. And since the matrix gene was from a unique lineage, we can differentiate those viruses that have picked up some of the pandemic genes through that matrix. So we look at both trends with subtypes, but also not all H1 are the same and not all H3s are the same. So if we look a little closer into the sequence data, we can break the H1s out phylogenetically into the alpha, beta, gamma, pandemic, and then the delta 1 and 2. And then with the H3s, we also saw these phylogenetic clustering with 1, 2, and 3. And then we had a long period where cluster 4 was fairly stable. But in the recent years, since we've got the reassortment with the pandemic H1N1, we're starting to see some of this cluster differentiation within the H3 as well. And again, using the matrix gene as a measure of reassortment between those two virus lineages. But we also are looking at the viruses subset at the whole genome level to try to understand the overall complexity of the viruses. So the next few slides were mostly generated by a former postdoc in my lab, Tavis Anderson, who's now on faculty at Georgia Southern, but he'll be giving his talk tomorrow. And then the new person taking over this work, Brian, has updated them for me. So basically what we're doing from the USDA surveillance data is taking the genetic sequences from the database and looking at where they break out phylogenetically. And this graph shows that we see trends. So earlier on in the surveillance system, the red is the pandemic H1N1. We saw this growth and detection of the H1N1, but then it rapidly kind of tapered off to where we don't see it quite as frequent or in a greater proportion as we do some of the other cluster types. Some of the constant themes that we have a relatively steady flow of H3 viruses, the delta 1 viruses, and then the gamma. So those have really stayed consistent throughout the system. And then we can see patterns such as the alphas, we have a blip and then it went away. And then we have this continual kind of percolating level of the H1 betas. So even though we've got kind of 3 dominant phylogenetic clusters, the others haven't gone away. They're still at risk of bubbling up and causing outbreaks again. And I'm sure when you look at sort of the system level, you guys are seeing that happening in real time. So this is the phylogenetic tree of the H3 viruses. And at the top here, we have the cluster 1, 2, and 3. So those are the historical viruses that were introduced around 1998. And then we had the cluster 4 that evolved from cluster 3. And what Tavis has indicated here in red are those H3s that have evolved forward into 2013. And so this is what I talked about earlier about now we're starting to see some of these cluster branches developing in this H3 cluster 4 group of viruses. And so all these viruses in red are co-circulating and potentially could be leading to some antigenic changes that will cause problems with the vaccines. When we get into talking about the human H3N2V cases that we've seen over the last few years, all of those H3s cluster down here in what we're calling a subcluster A right now until we see how things flesh out over time. But it is interesting to note that the H3N2Vs associated with the human cases fall out here, whereas we have all these other types of H3s that are out there in the pig population. And then our surrogate for reassortment is the matrix, and what we've seen over time, the blue is the trig or North American lineage matrix, the red is the pandemic, And we've just gradually seen the pandemic matrix gene taking over the endemic swine viruses, so that now about 75% of the viruses that we sequence have this pandemic lineage matrix gene. So whether it is contributing to fitness of the virus, and we're not really sure, but based on what we're seeing here, there is some advantage for our endemic strains to pick up this pandemic matrix gene. And so when we look at what's out there in the swine population overall, this is just one of the many variants that we have in the swine population, and that's what we've called here in Pravina Kittikun's paper genotype 1. So the pandemic H1N1 gene segments are color-coded by this lime green. So 1 gene from the pandemic plus 7 from the H3N2, 7 plus 1. So the nearly all of viruses coming out of the fair cases have been this genotype 1 H3N2 with the H3A cluster of hemagglutinin gene. However, recently this summer we've had a number of cases that have been this genotype 3, where in addition to the pandemic matrix, they've also picked up the pandemic PA. So time will tell to see if this particular genotype also continues to be a problem. But up till now, we were really focusing on this genotype 1. So one of the things that we did with the H3 viruses when we started recognizing this cluster differentiation was to look to see, are we seeing antigenic changes that go along with the genetic changes? And so this is some data that will also be in Pravina's paper that's been accepted. Looking at different H3 viruses and the antisera generated from those viruses, and whether we're losing any HI cross-reactivity. So green is good, yellow is pay attention, caution, and red is no cross-reactivity or significant loss in cross-reactivity. So basically what we can see across these new cluster types, which we've called A through E up till now, is that for some of the cluster types with some of the viruses, we still see pretty good cross-reactivity, but we're seeing a lot more red here than what we would have seen previously during cluster 4's relatively stable period. So I think it's something to be paying attention to, but time will tell which of these subclusters are going to be successful and hang in there, and some of them we've already seen pop up and then die out. So we don't know what's driving the diversity. Is it reassortment, pig movement, ineffective vaccines, or other unknown factors? But one thing we do need to pay attention to is, are our control measures working? And I think speaking with you over the last few years, we know that we've got a ways to go with getting effective vaccines that work in all age groups across the nation. And then we've also had the discussion a lot on, can we develop a better system for updating vaccines? And then finally, what are the implications for public health? So I'll go quickly through this because I think the other speakers will hit on this a bit more. But in 2005, it became a regulation that CDC needed to report any novel influenza A virus detected in humans to WHO. So we really started to see an increase in detection in people of these swine lineage viruses. So since then, we've seen a total of 366 of any subtype, but over the last 3 years, we've had 339 so far of these H3N2 variants that have this unique relationship with the show pigs and the fair venues. And so a lot of questions about why this is. I mentioned this before. They're all H3N2, cluster A, HA. They have a specific lineage of the neuraminidase, the 2002 lineage, and now we've got some with the PA from the pandemic as well. I think there's a lot of factors that are playing a role in why these particular viruses may be successful, but one of the obvious things is the work that the public health groups have done looking at the population immunity in different age groups of humans. And so this is actually the epidemiologic curve for the last couple of years. But so this is the serologic profile of the different age groups based on when they were born. And the bars that I've added here are just indicating those age groups that really have don't have cross-reacting antibodies against the swine H3s. So this really probably helps explain largely why we are seeing this in the kids and in a younger population exposed to the fair. So some future challenges, and I think this is my last slide, Jen. But we know that swine influenza A viruses in the U.S. are under a period of frequent reassortment, and this seems to be going hand in hand with hemagglutinin evolution. We need to do a good job of continuing to monitor these viruses and trying to interpret what is this genetic change going to mean for the antigenic diversity of our viruses. We've looked at some of the fully licensed products in our experimental models against these newer H3N2s. We've got a wide spectrum of efficacy against the H3N2 variant. And then one thing that's clear is if your pigs have maternal antibody at the time of vaccination, it did away with any partial protection that we had with those commercial vaccines. So that's a very big challenge for the industry, is the timing of vaccination for inactivated vaccines. We also have not had in the U.S. a uniform system for selecting swine vaccine strain updates. And although mass vaccinating has been shown to decrease viral shedding, it's going to continue to be difficult to manage mixed immune populations with vaccine. We don't have a sensitive and specific pen site test, and clinical signs aren't sufficient. Many of these H3N2 reassortants are pretty quiet in naive pigs, so they may not be showing much signs of illness. They may not spike a fever, but they're still shedding quite a bit of virus and transmitting it to naive pigs. Pig movement and mixing has been very difficult to track, so we really don't know the source of the virus, or I don't anyway. And there's been a lot of variability among these different venues for their requirements for pigs entering and leaving the fairgrounds. But to address this, there were some guidelines and recommendations that came out last year in time to implement for this year's fair season. So hopefully we'll hear from the other speakers on how well that has gone.